Oh, excuse me. Okay.
Like, when you sneeze into your
I'm
elbow.
recording right now. Hello, everyone. Sorry about that. Here we go. Getting started on my video for this Unit 2 essay number 1. To begin, we're going to talk about thick and thin muscle filaments and the anatomy of these muscles. And we'll start here with the sarcomere. The sarcomere can be easily identified when you identify two Z discs or two Z lines. It's usually found in between them, as I have noticed here. So everything between these two gray lines, like I've identified here with the Z disc, is a sarcomere. Well, let's dive in a little deeper. Right here we have the myosin, right here. The myosin forms the thick filament. The myosin also has myosin heads wrapped to it. And these are the means by which we contract muscles. We contract and shrinken these sarcomeres to contract our muscles, whether that be lifting weights and, moving our bicep that way, or whether that just be like picking up a cup of juice with our bicep and bringing it towards our face. As we move on, we see here, as I have identified green, I've identified these little guys as titin. Titin is what holds on to the myosin and is the means of flexibility within the muscles. It plays a big role in stabilizing it, and then when we do have contractions, it also shortens. And when we relax, it's like the one that really helps us kind of reestablish that. And then, as I identified here, the H zone is between our friends, the nebulin here. Nebulin on it usually has actin on it, followed by tropomyosin, followed by troponin, which we'll get into a bit. And the H zone is basically just the myosin here, and doesn't account for the nebulin and the actin and the tropomyosin on the side. Let's get into more physiology now that we've identified these things. Tropomyosin, calcium, and actin are three parts of the binding site of the troponin. So we have the T site, the C site, and the I site, and like I've shown you guys here, the T site, or the T site of the troponin, is going to attach to the tropomyosin, which we identified here, which is that thin, little thin form of stuff that sits on top of the actin and is preventing the troponin from, or is preventing the myosin heads from reaching them. So we need something that will attach to the tropomyosin. We need something that will then attach to the actin to pull the actin, or attach to the actin to stabilize. And then we'll use the calcium as our energy source. And that really is the last part that really gives us the green light on pulling that tropomyosin away. As you can see here, when we look at one strand, for example, here on the nebulin and the tropomyosin and the actin, when we look at one strand of actin like that, that is called the G actin. When we look at both together, that is the F actin. So troponin, like I wrote here, is going to peel tropomyosin off. Why? Well, I'll tell you why. So right here, I've diagrammed our myosin head here. It has ADP on it. Myosin is made out of a light chain and a dark chain. This is the the light chain and the heavy chain. This is the heavy chain, and the light chains will just hang out around. There's 4 of them. Light chain here. Heavy chain is the purple. So I just drew it here, but I just drew the troponin here, but that goes for each section here. But anyways, then we have contraction that's going to happen. And here's the means in which now we see the myosin heads attaching, because the troponin is pulling the tropomyosin off of the actin. So now, as you see, with calcium attaching to the C site here, it's going to then also allow the ADP to attach. When we attach the ADP, we're going to see heat get released, and the heat is also going to break off this phosphate bond. This phosphate is going to pop off, and this ADP is also going to pop off due to the attachment, which pulls the actin towards the middle, closer to the myosin. And this does it on both sides of the myosin here. So it's going to pull it in this way and this way. So it pulls it towards the middle. And the removal of the phosphate causes it to come into the uncocked position. And when we see this, it moving forward, we refer to that as the power stroke. It used power to move the actin towards the middle. Once we do that, we then see ATP attach to the myosin head once again. When you attach something, energy is used and released. So we see more heat bouncing off. And then finally, to get to back to square one, we see hydrolysis, which is turning ATP into a phosphate and to ADP. And this happens constantly till we pull the actin towards the myosin, forming that contraction. So this just happens again and again and again until we get that contraction. Cool. Now let's get into the means in which this calcium comes into the sarcoplasm to activate our friend troponin that we just talked about. Really quickly, because we should already know this from last unit, I'll just fill in some of the gaps. Action potential comes down, opens up the voltage-gated calcium channel. Calcium leaks in and allows acetylcholine to form in these vesicles. Snare proteins bring them down and release them through exocytosis. And the acetylcholine then attaches to these acetylcholine receptors, allowing ligand sodium channels to open up. Once the sodium comes in, it sends more of an action potential off, allowing the sodium voltage gauge channels open and let more sodium. Then we get this action potential heading down to our DHP, which is going to be like the action potential receptor, which is linked to the RYR here. That's good. Then going to allow calcium to leave through the sarcoplasmic reticulum into the sarcoplasm. We have our little friend here, calsequestrin, that's going to hold on to calcium near the exit area, just so this process is as quick as possible, and allows calcium to leave through the RYR into the sarcoplasmic reticulum at the quick speed that we need. This all happens. This section here is known as the sarcolemma, right here, where the postsynapse is right here. It's going to receive those neurotransmitters, and then we have the T-tubule here, which also holds on to which has those DHP channels. Next, we'll go into aerobic and anaerobic sources. Energy sources, I mean by that. So aerobic is basically respiration. It relies heavily upon oxygen. This is what really helps us get glucose and ATP throughout our body. It is slower acting, but it is very accessible. And then we have the anaerobic, which is even quicker, which falls into which we know is creatine, ATP, and glycolysis. And the speeds in which these are used are right here. Creatine 10 seconds, ATP 5, glycolysis 1 minute. Whoops. And then we're going to get into muscle twitch. Muscle twitch is just as follows. There's 3 different types. Type 1A, type 2B, and type 2A. My apologies. Let's go back here. This is the circuit pump here. This is what's going to pump calcium back into the sarcoplasmic reticulum. And this is used from our skeletal muscles. Skeletal muscles use mostly intracellular calcium, which is a bit different from our smooth muscle, which we'll learn a little bit later. So our circuit speed is different between all 3, as you can see here: slow, medium, fast. Type 1A uses aerobic, while type 2A uses both. And then type 2B uses the anaerobic ones we were talking about. Then you can see the amount of mitochondria they use. It really makes sense that aerobic has lots of mitochondria because it's producing and uses a lot of ATP. But what's interesting about type 2B muscle twitch is it uses few, because we rely on these other resources right here to perform those things. Then we get into the type of smooth muscle. So we see the single motor unit and the multimotor unit. It says, what we've prepared for you guys is one single contraction that moves in a wave is what we see with a single motor unit. This falls into our digestive tract quite a bit, just slowly moving things throughout our digestive system, contracting them down, so then we could get those nutrients we need and get rid of the ones we don't need. And this works completely independently of the nervous system. This is something we don't control. This is involuntary. Multimotor unit. These cells aren't electrically coupled like we see in the single motor unit. These require different stimuli to work down these different units for us to see these certain things happening in our bodies. So it really depends on the input from and information from our nervous and endocrine system. Smooth muscle contraction. So we have 2 different types: tonic and phasic. Phasic is more of a rhythmic. Rhythmic is the right way to say it, guys. Rhythmic contraction, which it kind of goes in cycles. It's one after another, and it's going to constantly contract throughout our body. Tonic is a long and usually, I don't know if single is the right word for it, but slow, constant contraction. And you see this a lot in sphincters. Sphincters are the things that help you not poop your pants, right? They're going to hold on to things when they need to be held on to, and when they need to get discharged, they will be. Let's move into the means in which calcium, how calcium impacts the smooth muscle of the cell. So first and foremost, we have our DHP channels, which are opened by depolarization. We have our G protein, which causes alpha protein to attach to the PLC, which is right here, which then causes an increase in IP3, and which then opens our RYR. So IP3 will bind to the SR in a way which causes calcium to be released, and like I said, that RYR will open up. So that rapid increase in intracellular calcium results in an increase of MLCK, which you see is caused by the attachment of calcium on our calmodulin MLCK complex. And then as we move along, we get into what we see here, the rhinokinase, which phosphorylates MLCP and will inactivate it. Here's MLCP, guys. It is activated or inactivated by the Rho kinase. When it's phosphorylated, it's inactive, and when it's not phosphorylated, it's active. So it's ready to eat. Sorry, it's listed differently on there. It has been shown, okay, and excuse me. My stomach hurts really bad. Okay, calcium will deactivate the Rho kinase. So when calcium deactivates Rho kinase, that means we're going to end up having a active MLCP. So as things depolarize, we see that there's a mechanically gated calcium and a ligand gated calcium. Mechanically can be activated by stretching, increased blood flow and all of that into our muscles, causing more calcium to be let into the mechanically gated channel. Then ligand gated can also be due to certain neurotransmitters that we receive. So yes, and smooth muscle is a 50-50, we kind of say, between ICF and ECF calcium that we use. So more ICF causes more MLCP, and we'll see MLCP coming along earlier in the cycle. We see this with the tonic right here. The tonic right here on the side, and that is I am forgetting the name of that. Oh, Crossbridge. Sorry, I didn't even need to look at my notes. I remembered. So this is the tonic Crossbridge, and as we see here, we start with an ADP, and then ADP on the myosin head with a phosphate. Then we see MLCK, which is kinase, right? So it's going to phosphorylate and put another P on the light chain. Then a reaction is going, once it attaches, and then the two phosphates are going to pop off. One off the light chain, one off the myosin, and MLCP is the one that's going to cause these phosphates to be removed, and the ADP pops up. Then we get into the uncocked position here. This is known as the latch state because when this ADP pops off, it kind of changes the shape of this myosin head into a weird position that makes it hard for ATP to attach and then return into our position here. And then the tonic is good, like we said, for sphincters and just a contraction to not let things pass through. Phasic is a little different because, like we said, tonic is when we have a large ratio, we have more MLCP than usual. And then we have phasic here, which is somewhat similar. It's activated by calcium. MLCK phosphorylates it, causing that ADP to attach. And then ADP leaves, and phosphate also leaves. But we keep the phosphate here that we received from the MLCK. And then we see that ATP is attached. But it's really interesting here is we don't always see it going this way. We can see through the phasic that hydrolysis occurs. What was it called again? My goodness. And that allows just the phosphate to reattach rather than going through the MLCP, which will eat up our phosphate, and forming hydrolysis here. So yes, here are the graphs related to the phasic and tonic system. Then we get into the chain fiber and the bag fiber. This is a section found within our muscle. It has a type 2A receptor and a type 1A receptor. Type 2A shows it still shows our brain and our nervous system that we're still stretched. And then type 1A is firing signals to let us know that if we're actively stretching. Dynamic gamma motor and the static gamma motor go to separate things. You can see static to the chain fiber. Dynamic gamma motor goes to the bag fiber. Here are some features of skeletal muscle versus smooth muscle. Appearance, skeletal muscle is striated. You can kind of see those striations within those big bodybuilders, right? Like, you can see those individual striations. Smooth muscle is not striated. Control, skeletal muscle is voluntary. Smooth muscle is involuntary. Contraction speed is fast and slow. Fatigue resistance, less high. Calcium source usually comes from the sarcoplasmic reticulum in skeletal muscle, and mostly extracellular and smooth, really 50-50. Regulatory protein, troponin and tropomyosin. Comodulin and MLCK for smooth. Next, we have our reflexes, which we'll talk about the Golgi apparatus reflex here. This is basically where our neurotransmitters and our neurons are connected to, let's say, our quad here. And we're trying to stretch it out. However, when we stretch something out, we want the other thing to be relaxed, right? Sometimes our body compensates and doesn't really do that. For some reason, it tries to compensate. And it causes a large tension on our tendons, which can be very dangerous. And once our tendons are strained, that could really mess things up. So we have receptors from our spinal cord here, and from our thigh, our quad here. What's really interesting about this is we're going to receive signals to our central nervous system, and it's going to be like, oh shoot, our tendon is stretching quite a bit. Then it will send another signal back from the spinal cord telling one muscle to be inhibited and one to be excited so that we can take that strain off of our tendon. So we see that here. We have the IB, which tells our thigh to be inhibited, and our signal received from the tendon tells us that we are stretching too hard, the tendon that is. And then our spinal cord tells our other muscle, which is our hamstring here, to then contract to take that terrible strain off our tendon and protecting it. And here, with our neurons, we have interneurons, which are neurons between two neurons, and they are going to communicate, like for example here, to inhibit or sometimes to excite certain muscles. So they just help that information get moved along. That is our video.